and I am back with the cell 2. I apologize for the lighting in here. It is getting later and later as I record these, and so the lighting is just getting crappier and crappier. Anyway, the cell 2, let's go ahead and work on that. Okay, pre-lecture assignment. So there was quite a bit of vocabulary in the pre-lecture assignment for this. As always, stop and go back if you haven't read, because honestly, you're going to have an impossible time with the rest of this packet if you haven't read this first. And that's just because of the amount of independent work, if you will, that this packet contains, where you kind of have to think through and apply what you've learned in large part from this page to what you're going to see in the questions that get asked. So let's go ahead and start covering our pre-lecture assignment. Okay. The function of the nucleus is to store genetic information. That's what it's there for. Okay, I think your book says store genetic info as long strings of DNA or long chains of DNA or something like that. The nucleus has different parts to it, as you can see, including something called the nucleolus. Okay, it's located inside the nucleus itself, and this is where components of ribosomes are made. Ribosomes, okay. And then we have something called the nuclear envelope. That separates the nucleus from the cytoplasm, and it has nuclear pores. Sorry, I just skipped ahead one. It is continuous with this is the endoplasmic reticulum. We'll go over this in a second. Ribosome proteins move into and out of the nucleus through nuclear pores. Okay. You can't see it super great, but in this image, again, you've got the larger nucleus itself. Here's the little nucleolus where the ribosome components are made. Those ribosome components, and also some of the genetic information, is going to move out of the nucleus. Ribosomes are going to leave the nucleolus and go into the nucleus, and it's all going to leave the nucleus through these little holes here, which are the nuclear pores. Okay, the nuclear pores are in the nuclear envelope, which is essentially the membrane that's around the nucleus. And then you can kind of see it here, but these little, look like little roads or little highways over here, this is the endoplasmic reticulum, or at least part of it. And it is usually attached to the nuclear envelope, because the stuff that's going to come from the nucleus is going to need to be assembled, or needs to be finished being assembled in the ER, or the endoplasmic reticulum. Okay, so that's the nucleus. Then when we get to ribosomes, ribosomes are composed of protein, a macromolecule, and something called R, or ribosomal RNA, which you probably remember. Actually, we haven't gone over that yet. rRNA. So RNA is another macromolecule. It's a nucleic acid, and this specific type is called rRNA, and the R stands for ribosomal. Their job is to make proteins. So they're really important, which is why even prokaryotic organisms have them, because if you have genetic information, but you don't have any way to build the proteins that come from that genetic information, you kind of don't have life. So the ribosomes are even in these prokaryotic organisms so that proteins can be built. Ribosomes can be found either free-floating in the cytoplasm or attached to the endoplasmic reticulum. Either way, their job is the same as far as protein synthesis, but the only thing that really differs is kind of what those proteins are going to be used for, okay? So there's two types of endoplasmic reticulum, kind of collectively called the endomembrane system. The rough ER is studded with ribosomes, which is why it's rough. It looks bumpy. Okay, so from here, proteins are going to be packaged into vesicles, which we kind of talked about what vesicles were in the last lecture. Okay, and then we have smooth ER. Smooth ER does not have ribosomes attached to it, and its job is to synthesize, I'm going to put lipids here, as well as phospholipids. So we're looking at lipids like steroids and phospholipids, part of the cell membranes. So that's what the smooth ER is going to be responsible for producing. It doesn't really talk about it here with the rough ER, but the rough ER tends to make things that are protein rich, like enzymes, stuff like that, okay? Then we have the Golgi apparatus, and the Golgi does like everything. So we have these production centers, right, where we've got the nucleus with its genetic information that can be copied. We have the nucleolus with its ribosome parts. We have the ribosomes themselves that make proteins. We have the rough and the smooth ER. But then we have the Golgi, and this is where stuff is going to get processed and packaged and then secreted. Okay, the Golgi is also going to store those things until they're ready to be secreted. So that it's going to store them until it's ready to go. I always think about a Golgi as like a warehouse. You know, the stuff that's been manufactured all comes into the warehouse where it gets processed and packaged, stored until it's time to ship, and then it gets shipped, or in this case, secreted. Okay, then we have lysosomes. Lysosomes are kind of like little stomachs in the sense that they are organelles that are nothing but sacs of digestive enzymes or hydrolytic enzymes. Okay, and they break down foreign matter. They not only break down foreign matter, but they break down waste products in the cell to keep the cell clean, essentially, from having unwanted parts building up. They perform something called, I can get my pen to work where it should, autodigestion. I'm going to write it down here so I have room. Autodigestion, where parts of the cell are broken down, okay? So that's what I was just saying as far as kind of the waste or the scraps left over from building these other materials in the cell, okay? There's a couple of disorders around lysosomes that you may get to talk about, like I said, getting late, talk about in our genetic section. Okay, we move to the next page. So you've read about all these different organelles. We've talked about the main points for the main guys. And now we're going to go into a little bit of detail about the cytoskeleton. So we've got this membrane that goes around the cell and kind of keeps everything in that's supposed to be in and out what's supposed to be out. But we also have something that provides some structure to the inside of the cell. And that's the cytoskeleton. Much like our skeletal system is packed inside our body, a cytoskeleton is packed inside the cell. It's this protein scaffold that provides structure for the cell and helps to maintain its shape. It's not rigid like you think of bone as being rigid. It still has the ability to bend and shift and have its shape changed a little bit as the cell moves, if you will, or is exposed to the conditions of its external environment. But it does help to keep the organelles in place. It helps to keep the organelles from, I don't know, all falling to one side. Not that they would, but in theory, right? It keeps them in place. So as part of our cytoskeleton, we have these three components. We've got microtubules, actin, and intermediate filaments. Okay, so I'm just gonna write down here what each of these do. Microtubules are going to maintain the cell shape. And again, within reason, this isn't crazy rigid. That wouldn't be good for the cell because the cells are under pressure or they experience changes in their environment. And if they were super rigid, everything would just crack and break. Microtubules also provide kind of like a highway system for vesicles. And if you remember, vesicles are those pinched off parts of membrane that transport foods and liquids in and out of the cell. Okay. And microtubules are also extremely important in the process of cell division. When it comes to mitosis and meiosis, they're the ones that are going to actually pull chromosomes apart. Did I just really... It did. I just spelled it cell division. You guys, oh my gosh, I'm like tired. Okay. Oh, crack myself up. I don't know if that's a good thing though. And I'll make that look more like cell division. How's that? Is that better? I know you can relate. I know you can relate. It's called recording lectures at night. And the kids can't come running in or like be screaming hysterically in the background. Okay. Anywho, microtubules. These guys originate from a centrosome, which we'll talk about a little bit more when we get into cell division. Now let's talk about actin. So actin is going to kind of line the edges, the periphery of the cytoplasm. Cells don't walk, but you can think about the actin as kind of allowing for some of the cell movement, cells to shift from one location to another. And so you can think about it as cell walking. It allows for cell walking. Actin strands actually slide, how do I want to put it? They slide not really against each other, but kind of in relation to each other to achieve movement. I don't know why you would ever do this, but in case you want to try. If you're to lay down on the floor, on your stomach, with your arms at your sides and your legs together, you could kind of think about, you know, if you were to shift one hip forward and then the other hip forward, this constant shift back and forth would allow your body to move without using your arms or legs. Kind of like how a snake's ribs work for movement. It's not exactly the same process, but in this moving relative to each other, it can kind of give you a picture of how actin can allow a cell to be able to shift or to move position a little bit. Okay, then we have these intermediate filaments, and these are going to provide structure for the nucleus specifically. Okay, and they also are going to be used to tether cells together. Obviously, cells are all together in tissues, right? So you have a bunch of skin cells in close proximity to each other on the back of your hand, okay? Well, something has to hold these skin cells together. They're not just a bunch of individual random free-floating cells. They have to work together to fight infection, to heal, to send nerve impulses, things like that. So what holds them together are these intermediate filaments. So when we're looking here then at this diagram, this has been colorized. So these are 4 cells in close proximity to each other. And again, this is not the color that they actually are. These types of micrographs come out usually black and white and gray, usually black and gray. But this has been colorized so we can distinguish what is what. So these green areas here, these are going to be the microtubules. And again, this is that kind of highway system for vesicles. You can see that it's concentrated largely around the nucleus, which makes sense because that's where a lot of our manufacturing is taking place, is in this area. Then we have all of this white out here around the edges. Well, if you remember what is around the periphery, around the edge of the cytoplasm, that's going to be the actin. And it's appropriately located to be able to allow movement. This blue turquoise color, these are the intermediate filaments that are supporting the nucleus. Okay. Nifty. Okay, so this next part is classwork, except we're not in class together that way, are we? No, we're not. So as always, for your own understanding, and even to test your own understanding, pause it here and please read through these and see if you can identify which of the 3 cytoskeletal components we just talked about, the intermediate filaments, the microtubules, and the actin, which one of those would be responsible for these different abnormalities or these different disorders that are being discussed here. So I'm going to assume that you're going to pause it and use what you just learned about these proteins to decide which one is responsible for these issues. Okay, now I'm going to assume that you came back. So let's look at this. Epidermolysis bullosa, abbreviated EB, is a group of rare inherited skin diseases that are characterized by the development of blisters following minimal pressure to the skin. So light touches and you get blisters. Blistering often appears in infancy in response to simply being held or handled. In rarer forms of the disorder, EB can be life-threatening. There's no cure, and treatment focuses on preventing and treating wounds and infections. This one is, I don't know if it's tricky, but it definitely makes you think for a minute about the possibilities here and then the different applications of these cytoskeletal components. So what happens, what we're looking at here are actually the intermediate filaments. I'll walk you through that. So remember that intermediate filaments are responsible for kind of holding cells together, right? Well, in blistering, you kind of have this separation, if you will, of layers of skin. They often fill with a fluid that can be infected. And so in this particular disorder, it's the intermediate filaments that are having the issue because they are not holding those skin cells to the epidermis. Okay, so the deeper cells in the skin aren't being anchored to the epidermis, which allows these layers to form, or the separation of layers in the skin, and for these blisters to form. Okay. All right, let's look at the next one. Metastatic cancer cells use cytoskeletal bundles to underpin protrusions that allow them to break away from a primary tumor and invade through the surrounding tissue. Okay, cytoskeletal bundles to let them break away from the larger tumor itself, as well as the tissue surrounding the tumor. After traveling in the vasculature or lymphatic system, this is the lymph system that drains fluids, or vasculature is through the circulatory system, blood flow. So after traveling through that, they exit into a new niche and seed a new tumor. So now you've got that tumor cell that was in your liver, and it's moved through the body after breaking away from the tumor and starts a new tumor in the lungs. They exit into this new niche, seed a new tumor, often lying dormant for months or years, okay? During metastasis, cells adapt to their motility and adhesive capacity to suit their environment in much the same way as embryonic cells do during morphogenesis. In other words, how a cancer cell creates a tumor in the liver is going to be different to how it's going to create one in the lungs, because the environment and the tissues are different between those two organs. That's what happens in embryonic cell development. Depending on where the cell is, the stem cells are going to get triggered to create different organs. Okay, now that we've gone through all of that, this one had a lot of medical terms in it. We're talking about movement, right? We're talking about cells' ability to move from one location to another. So what we're talking about is actin, kind of allowing that to happen, allowing cells to break away from the larger group through the cell walking, okay? Paclitaxel, actually, I don't know if it's paclitaxel. I'm gonna call paclitaxel. Sold under the brand name Taxol, is one of several cytoskeletal drugs. Paclitaxel-treated cells have defects in mitotic spindle assembly, chromosome segregation, and cell division. Paclitaxel stabilizes the protein polymers and protects it from disassembly. Chromosomes are thus unable to achieve a metaphase spindle configuration. This blocks the progression of mitosis, and prolonged activation of the mitotic checkpoint triggers apoptosis or reversion to the G0 phase of the cell cycle without cell division. Not a great thing. It just is saying that because it doesn't get to the point where we have the mitotic spindle assembly, chromosomes can't be pulled apart. What pulls chromosomes apart? Microtubules. Okay. Alrighty then. Cytoskeletal structures. We'll talk about cilia and flagella. So cilia and flagella are other specialized microtubules that can allow for cell movement. Cilia actually comes from the Latin word eyelashes, which kind of looks like, right? They move in kind of this back and forth motion. I like to think about them as windshield wipers or oars in a boat. So they're not like all flying everywhere in different directions. It's like all going together one way or the other. In the body, one of the places that you find cilia is in the trachea and in part of the respiratory system. The cilia in these locations, which are part of the cells on the outermost layer, they act like a broom. Okay, so we've got windshield wipers, the oar motion, or like a broom sweeping back and forth. And that's what they do. They sweep out foreign particles. So, you know, obviously in the process of talking and breathing and being alive, you inhale all kinds of things. We don't want most of those things in our lungs. Well, the trachea and the respiratory system have these cilia that help to sweep that back up towards the mouth where it can be coughed out, and doesn't let it go into the lungs. Okay, so all right, then we have flagella. Flagella, also microtubules. These form more of like a whip shape. And they function like a whip as far as the motion. In the human body, the only place you're going to find flagella is going to be in sperm. So you don't even see it in the female body, you see it in the male body. And that's the only place you're going to find it. Which leads to this interesting question. How fast do sperm cells move? So, if you have ever been wondering about this, today's your lucky day, because you're going to find out. So essentially, if you were to picture this microscopic sperm as being the size of a salmon, it's a decent sized fish, it would be moving at the equivalent of 500 miles per hour. That's one fast fish. So if sperm, first off, that's a horrifying thought, if sperm were the size of salmon. If sperm were the size of salmon. They'd move at 500 miles per hour. Oh my gosh. Okay. So that's how quickly they swim as an equivalent. In the context of a human body, they can move approximately like 5 millimeters in a minute. They don't have terribly far to travel in that short space. So that's the same as if they were able to move 5 entire body lengths in a second. And again, that's adjusted because the space between the vagina and trying to get to where an egg is being released is not that far. It's obviously not a body length. And they're not the size of salmon. They're just kind of to help you put that in context. Okay. Oops. Let's try that again. In-class problem set. Pause here. The information that you're going to need to be able to answer these is going to be kind of back on that first page, actually, the pre-lecture assignment, where it's going to ask you or let you know what the different functions of the different organelles are. So take what you know about those organelles and what they do and apply it to the functions of these cells that they're giving you. It may seem intimidating at first, but it's really not. You have the information you need to at least make a good educated guess. So the first type, Leydig cells, these are going to make and secrete testosterone. Okay, so if you're looking at your pre-lab assignment, we have the information that we're going to need for this. Okay, so what is testosterone? Well, testosterone is a steroid. Well, what's a steroid? It's a lipid. Hey now, what's going to be part of producing lipids? That would be the smooth ER, right? So something that's going to produce a lot of lipids, something that's going to produce a lot of these steroids, then it's going to have a lot of rough ER, or rough. It's going to have a lot of smooth ER, because that's what smooth ER produces. Salivary glands make and secrete enzymes into the mouth that break down starch. Salivary glands, again, have these enzymes, and enzymes are proteins. What didn't completely get covered in the pre-lecture was the fact that rough ER is responsible for making a lot of this. If you put ribosomes, I'm good with that. In the sense that you know that ribosomes are going to be making proteins and that enzymes are proteins. But really it's rough ER. White blood cells destroy foreign material within the body. These are lysosomes. If you're going to be doing a lot of destroying and a lot of breaking down, you're going to need a lot of lysosomes because that's where the destroying happens. Muscle fibers that contract. What are you going to have a lot of? This one isn't really from your pre-lecture assignment. It's more from the cytoskeletal stuff that we went through. When you think about muscle contraction, it's this process of things moving against each other, and that's where we're going to have our actin. Okay. I don't know why I just did that. I just gave you a double thumbs up. I have no idea why, except it's late, and I guess I'm tired. But I've said that a couple times. Good news is we're done. We got through both of the cell, and so you've kind of got what you need for this week. And as always, please email me or drop a message on one of the discussion boards. If you have questions about any of this material, if you're not comfortable with the material, if there's something I went over that you're just not understanding or that I didn't cover very well, please let me know so that I can get you prepped for an exam that's coming up soon. Okay? All right. On that note, I will talk to you soon.